Hi, my name is María Farfán and my friend's name is Escudero. Today we are going to talk about occurrence of virulence genes in multi-drug resistant E. coli isolates from humans, animals, and the environment. One health perspective.
So, what is Escherichia coli? It is a gram-negative, rod-shaped bacteria that lives in human and mammal intestines alike. While most strains of it are harmless, some of them that can be consumed through fecal matter directly or through contaminated food or water can cause symptoms such as stomach cramps, diarrhea, and even kidney failure.
Now I am going to answer this question. Okay, bacterial resistance occurs when bacteria develop mechanisms to survive antibiotics, making them ineffective. A more serious level is multi-drug resistance, where bacteria resist several types of antibiotics, limited to treatment options, which represents a danger to public health, and these bacteria can cause infections that are difficult to treat and contribute to the spread of resistant genes, increasing the risk of intractable disease in the future.
As for the main objective, it consists on testing the occurrence of virulence genes in multi-drug resistant Escherichia coli isolates from humans, animals, and the environment.
Okay, now method. The study used the Dix diffusion method of Kirby-Bauer to assess antibiotic resistance in E. coli isolates and confirm multi-drug resistance. Additional multiplex PCR was able to detect virulence genes, analyzing their relationships with bacterial infection potential. DNA extraction is crucial in microbiology and genetics. This study used the boiling method, heating bacterial cells at 100 centigrade for 10 minutes to lyse them and release DNA. After the centrifugation for 10 minutes, and DNA supernatant was stored at minus 20 centigrade for later use.
Unlike standard PCR that uses primers targeting any DNA sequence of interest, the PCR mixture for the detection of virulence genes amplifies all the DNA sequences related to bacterial pathogenicity genes. When it comes to materials, it doesn't differ much. This type of PCR is crucial as it enables a rapid detection of virulence genes in E. coli strains from humans, animals, and the environment, helping to explore how these genes coexist with antibiotic resistance by comparing both types of genes within the same bacterial strains. Gel electrophoresis. It is a technique that separates DNA fragments based on size and charge. A DNA sample is loaded into a gel, and an electric current is passed on, causing the negatively charged DNA towards the positive end. Smaller fragments travel faster, allowing for separation by size. In the PCR context, gel electrophoresis is used to visualize amplified DNA products. The separated bands on the gel indicate the presence of target genes, such as virulence genes in E. coli. This method confirms how virulence genes coexist with antibiotic resistance genes, providing insights into potential risk for infections and treatment challenges.
Okay, the bar chart shows the prevalence of some genes in multi-drug resins, E. coli for humans, poultry, pigs, and the river water. High frequencies appear in the human and birds, while EA is lower in pigs and water, and STX1 is absent in all samples, indicating multi-drug resins E. coli carriers, multiple virulence genes, posing infection risks. The electrophoresis health shows 15 lanes, with L as the molecular maker, N and P as negative and positive controls, OMPA and TRAC appear in multiple samples, both in lanes 3, 6, 10, 12, 15, only OMPA in lane 1, 2, and only TRAC in lanes 4, 5. This suggests variety distribution of virulence genes.
In Figure 3, there are two genes mentioned. The first being the EAEA gene, shown in lanes 1 to 5, indicating E. coli strains with adhesion-related virulence, also called the bacteria's ability to adhere to host cells. The absence of the second gene, STX1, suggests these isolates are not enterohemorrhagic E. coli, where there would be bloody diarrhea. Lane 6 is negative for both genes, confirming variability in virulence markers. In Figure 4, the lanes 1, 2, 4, 5, 10, 11, 13, and 14 carry the BFP gene, linking them to enteropathogenic E. coli that causes watery diarrhea without the use of any toxins, while its absence in other lanes highlights genetic differences in the types of sickness this specific strain causes.
In this discussion, the table presents comparisons between the findings of the study and other authors who have investigated virulence genes in multidrug-resistant E. coli. For example, firstly, the study coincides with this finding, so it is approvable. Nimnoi, the review article compares this percentage, so it is also agrees. And Nielsen, since the study found the presence of OMPA in multiple isolates, it goes valid.
Jumping to our conclusions, our first one is that the study presents how resistance and virulence genes coexist, stressing the need to improve our antibiotic strategies to combat multi-drug resistant infections. The second tells us that PCR and electrophoresis help track virulence genes, improving disease surveillance. And finally, the presence of genes in humans, animals, and environment proves the need for cross-disciplinary strategies to control our infections.